Hello. Today I wanted to talk about the effects of atrazine in human cells because I've been seeing in the news cycle, especially with RFK and his posse making their rounds, that atrazine was floated as this very dangerous chemical in our water and food supply that was very toxic, and in one instance, they were saying had feminized frogs or reversed their sex to some degree. So I wanted to do my own experiment and see what the data had to say about atrazine. So atrazine is actually a herbicide used in the United States for like corn and stuff, particularly in the Midwest. And this has been banned for 20 years in the EU because they found that its concentrations in the groundwater exceeded what is considered safe. Um, and that was because the manufacturer could not show that these levels were safe. Atrazine has been detected in our water since 2001, but the EPA claims that the concentrations are not harmful. And there was an ecological risk assessment in 2016 that said that the effects are still hard to determine, especially whenever you consider that the concentrations are very, very low in the water. So these are some maps I found that show the estimated use on agricultural lands as well as the predicted concentrations in the water supply. And as you can see, there is quite overlap between the usage and the predicted concentration, as you would see. But it's hard to make sense of these numbers if you don't know the effect of what this is causing. Like 63 micrograms per liter, or I'm sorry, pounds per square mile, could be quite drastic if this herbicide is quite potent. So we need to really assess the effects. Also, to note, the concentrations of atrazine are present in many states, but in very, very low quantities, like 0.03 micrograms per liter, which is barely anything. It's in the parts per billion level. So how does atrazine actually work? Um, Again, it's a herbicide, and its role is to inhibit photosynthesis in plants. So if we have a house plant, all living things are composed of cells, particularly plant cells. What differentiates a plant cell from a human cell is that plant cells have cell walls. They're more rigid and can support the plant structure, but they also have these things called chlorophylls. They are um, stacks of light absorbing um, biological material. And what atrazine does, this chemical inhibits this PQ pull and prevents electron transporting within the cell. It's very important for energy purposes in the cell to have electrons flowing in the chloroplast, and it uses this to make glucose. If you disrupt this effect, you get a lot of oxidative damage and you starve the cell. And the effect is that the plant wilts, but your healthy plants live on, and you don't have to manually go into your field and pull weeds, essentially. So, what I want to ask is, how does atrazine influence the human cells? So, there is this repository of cells which are treated with all sorts of compounds. It's called the connectivity map, and it's by an institution called the Broad Institute. They like to host a lot of genetic data for that, and of the particular cell lines that I could find, these are cancer cell lines, particularly what I could find was a breast cancer cell line, MCF10A. And, why use cancer cell lines when you could use normal cell line is that if you took one of your cells and tried to grow them on the petri dish, it would grow and divide for a little bit, but then it would die. Because the reason you're not riddled with cancer right now is because your cells have a fail-safe. That cell would say, "Oh, we're dividing too much. We should shut down operations," and it'll die. But a cancer cell lacks that switch, and it'll keep dividing. So it's really used in this case for experimental purposes and to make the screening process possible, and that's used in the pharmaceutical industry to ensure safety as well. But in these MCF10A cells, we're going to be looking at about 12,000 landmark gene expressions. And why look at gene expression? There's this central dogma of molecular biology, that's what it's literally called, where gene expression is actually a dynamic process based on a cell's needs. So we all know that we have DNA, and that makes us us. And that DNA is converted into another molecule called RNA through a process called transcription. It's like making a copy of a book and giving that to someone. You're not destroying the original copy. You could do whatever you want with the copy, read it, make paper airplanes out of it, whatever, but it doesn't harm the integrity of the original. And that RNA can become proteins or be used for regulatory purposes. But what we're interested in is comparing the abundancy of RNAs of different genes between samples to tell us how the cell is responding to a treatment. This is called differential expression analysis, and this is used in modern transcriptomics to really dial in how cells are reacting on the molecular level to various treatments we provide. So there's also some caveats when using this data. So atrazine treated cells exhibit a low range of transcriptional activation. So in this database, there's a score called a transcriptional activation score. And what this does is it assesses how strong or how pervasive the transcription is within the cell. So the larger the number, the more genes whose expression changed, the smaller, more genes were not affected by your treatment, right? So if something is quite toxic or quite effective, like doxorubicin, you would expect to have a higher TAS. It's also a marker of replication strength and signature strength for your data. But it's an important caveat to note that for the 6 samples that I could find across 3 different dosages, the max TAS was about 0.19, which is very low on here. So I don't have the replication correlation on the x-axis there. However, we can assume that this compound will either be inert at these concentrations or have subtle or reproducible results. So in order to really assess how these genes are moving, it's a very complex system. It's a whole symphony of expression. There's multiple moving parts, and old school genomics really focused on one gene being broken or mutated, and that would have one effect. Like in sickle cell or cystic fibrosis, those are one gene diseases, and you're able to track that to the disease. However, whenever you give a drug, there's many side effects. Like if you look at the bottle for ibuprofen, it'll treat your pain, but it'll also give you many side effects, and that's because small molecules aren't as specific as they should be. But in order to assess that, we're going to look at whole pathways to see their effect in expression changes upon treatment. Genes work together in pathways to perform specific tasks in the cell. And if you look at the left here, I have a pathway for glycolysis, which converts glucose through a bunch of different intermediates into pyruvate. And this is very central to creating energy for your cell. And if you notice, this is a chain of events, right? So if you break one part of this chain, then you're going to disrupt a whole pathway, and it will have downstream consequences. Like if you mutate hexokinase, you're not going to be able to produce glucose 6-phosphate. So there's no reason to make aldolase, endolase, pyruvate kinase, and you'd see downregulation of all of those genes. And that's what I'm talking about, a whole system-related effect. And examining changes in expression within a pathway allows us to see these broader effects on the cell's phenotype, or what's physically happening to the cell. This technique that I'm using particularly is known as gene set enrichment analysis. And one particular pathway I'd like to introduce before we get into the data is the electron transport chain, which produces most of the cell's ATP. You can think of ATP like gasoline, and the cell the car, except in this example the cell is making its own gasoline. And it does that through a process called oxidative phosphorylation, where you have parts of the Krebs cycle that feed into the electron transport chain to shuttle electrons into this big structure on the right here, ATP synthase. That is what makes a majority of the ATP in our cells. So it's very important to have all of the stuff on the left coupled to the stuff on the right, that ATP synthase, right? It should really flow like water through a pipe, very elegantly. If you have too much water flowing through the pipe, you're going to destroy the right. Or if you decouple them or break their synergy, that's when you have very bad metabolic diseases, or you can make someone sick, for instance, if your drug happens to disrupt this harmonious action here. So whenever we get into the data and we look at the pathways, we can see that atrazine, in very high concentrations, cellular respiration was severely impacted. These are biological processes, very wide. So oxidative phosphorylation, aerobic respiration, cellular respiration, they're all just saying the same thing. These pathway genes are increasing in expression in a very large proportion, which is interesting. If we look further into what's happening at the molecular level, we can see NADH dehydrogenase activity is increasing and ubiquitin activity is increasing. Those are two particular enzymes involved in the electron transport chain that I was showing you earlier. They're like shuttles for the gasoline. And furthermore, if we talk about cellular compartments, the mitochondrial inner membrane and respiratory chain complexes and all these membrane parts are also being positively enriched. So at very high concentrations of atrazine. So what this means is that there seems to be some metabolic discrepancy caused to the bioenergetics of the cell. And what that means in fancy terms, or more simpler terms, I should say, is that um, the synergy between what I was showing earlier with the electron feeding complexes and the ATP synthase complex is being broken by this compound at high concentrations. And we can see this if we look at genes which are positively correlated with dose, right? We can see a dose-dependent fashion where the more atrazine you give, expression increases for many key genes, like UCP3 is seen to be positively correlated with atrazine concentrations. And this uncouples phosphorylation from ATP synthesis. Mitochondrial homeostasis by ROT1 is also being affected. There's pro-inflammation occurring at S100A9 and FOXP3. And another interesting factor I'll talk about later, but GAD45G, which senses DNA damaging agents, is also increasing relative expression level. So this isn't painting a very good picture for the safety of atrazine. And if we look at negatively correlated, so if you increase the dose, the expression should go down. You see CoA7A, which helps assemble part of that electron feeder complex, and oxidative phosphorylation is going down. The Krebs cycle is being impacted. Oxidative phosphorylation functionality by NDU-FB2 is being impacted. GSTK1, which is a scavenger for reactive oxygen species, which I will talk about very shortly, is also being impacted. And two sensors of DNA damage like HUS1 and SMC6 are being impacted, and a marker of the cell cycle, CDK7, is also being impacted. So what we can infer from this is that these positively impacted dose-dependent changes are uncoupling oxidative phosphorylation from ATP synthesis, and the cell's kind of freaking out because something is causing DNA damage. And DNA damage is especially worrisome in our cells because, as I was talking about with the books earlier, you can keep making copies of the book, of an original copy, without losing quality. But if you break the original copy, that information is lost forever. And that's how cancer cells can develop. If you keep destroying the original copy, the cell becomes worse and worse until it can't regulate itself and it turns cancerous. And especially with the downregulation of GSTK in a dose-dependent fashion, though it's hard to especially see from this data given the wide degree of deviation. Those are those error bars you see. There's something going on. It's not particularly evident from this data, but something in scavenging reactive oxygen species is perhaps stalling the cell cycle, and the cells might be trying to function with scavenging or sensing DNA damage. So it's not a very good picture for atrazine at these high concentrations. These are still like 100 times or 10 times what is present in the water. Also, as I was talking about before, there were claims made that atrazine was able to feminize male frogs to some degree, and I don't know if this data is particularly conclusive to that. However, you have genes like 19A1, which essentially creates estrogen, and genes like SRY and SOX9, which create males. And across all of these genes, there's no significant correlation seen between the dose level and the expression of these genes. So I don't know if this is particularly strong because, again, these are in breast cancer cell lines, which are originally female cell lines. Perhaps these would be stronger if the cell was derived from a male cancer cell line. But in this case, it's not particularly striking that atrazine caused more feminization or an enhancement of the female phenotype. But again, the toxicity of atrazine may increase reactive oxygen species. And what this is whenever you decouple the ATP synthase from the electron transport chain, these electrons that are fed in to the transport chain have nowhere to go, and oxygen will suck them up, and they'll suck them up one at a time, and they have nowhere to go, and they're quite volatile. Oxygen doesn't want to have this lone electron, so it'll just bind to anything and cause DNA damage or a wide degree of bad effects you don't want. Our bodies do have glutathione to combat this, but if it is being deregulated or it's being severely impacted and not replenished by the cell, that's quite a big issue, which could compound. SOD1 was also downregulated in the data that I observed. So in this very low quality figure I found on the internet, ROS can contribute to cancer cell development and cellular stress. Again, these reactive oxygen species, beyond protein oxidation, that's when the reactive oxygens bind to other proteins, or enzyme degradation and lipid peroxidation. The big effect is the DNA damage, which can cause cancer in normal cells, and that should really be averted, especially on a population level. Increasing cancer rates by having this chemical leak into the water is obviously not a good thing and should be prevented. However, although I'm showing that atrazine could be a possible carcinogen for its predicted ability to cause high levels of ROS in the human cells, you need a really high concentration of that, like 100 times the concentration of what I'd shown earlier is present in the water. And the acute toxicity, so if you just drank like a glass of it, is very low. You need a log of it to hurt you. That is the acute oral. Like, you need greater than 500 to 5,000 milligrams per your body weight. So again, atrazine concentrations are extremely minimal in the environment. Again, the likelihood that atrazine will exceed drinking water standard in shallow groundwater is very low, like 1% to about 10%. I don't have a date for this chart on the right, but it's really only a concern if it does increase like maybe 10 or 20 fold. Especially in the graph on the left here, there should be some counties in red where the concentration of atrazine is greater than 50 micrograms per liter. However, you will only get the effects that I had shown earlier, theoretically, if the atrazine is far, far greater than that. Like 50 micrograms per liter is only 232 micromolars. And the concentrations I'm working with again are like 33 micromolars or 100 micromolars. So it's not as dangerous as the concentrations in the study are shown. And I'm not particularly sure why there is a lot of this fear mongering about this very trace compound. I'm not sure about how well the regulators are keeping up on this, or if these charts are accurate. I know that they're a little dated, but there there's obviously the concern, like, oh, I don't want to be taking a carcinogen. It shouldn't be in the environment at all. And you're saying, yeah, it won't kill you, but I still don't want it, which is valid. And if there's something better, um, the farmer should certainly use something which has zero likelihood of causing cancer over something dubious. But you know, it's it's better than having like um, weed-ridden fields, which will drive down crop yields and increase prices. If you look at it from a macroeconomic model, but instead of arguing that, I'll just stick to the data that I showed. Um, so if you have any questions, please feel free to write them in the comments, and thank you for watching.